For Brain Awareness Week, I would like to present a story of the path that I took to my most cited paper in Neuroscience. I understood this when I made my Google Scholar profile and was surprised to see that this paper has the most citations. And serendipitously, it is the paper that was associated with a lot of hurdles along the way. So, I will discuss this. First, in 2004, I found myself searching for a job. Until that point, I had been studying the changes in glutamate receptors, NMDA receptors, and NMDA receptor-associated postsynaptic density proteins, and glutamate transporter-associated proteins in different brain regions from patients with neurodevelopmental disorders, namely schizophrenia, bipolar disorder, and major depressive disorder. And while there were some changes, as I found using either in situ hybridization or Western blot, these changes were not so great, and they couldn't account for all of the behavioral abnormalities that we observe in patients. And something else was important, the functional processes that these proteins participate in, such as synaptic plasticity. And I wanted to study AMPA receptors, which are more dynamic, but my mentor at the time thought that my skills and efforts are better to be directed to NMDA receptors. And he mentioned Dr. Rob Malenko to look up his papers. And I looked up the papers of Dr. Malenko on synaptic plasticity and started reading and understanding about this area. While searching for a job in the science careers, I saw an ad for a fellowship for people like me, medical doctors who wish to be involved exclusively with research. The requirements for applying for this fellowship were to have passed the USMLE Step 1 and 2, to have some background in research, and to contact one of the possible mentors faculty from Albert Einstein College of Medicine Department of Neuroscience to discuss a possible project and training in their lab. There were many excellent faculty, and I was wondering what approach should I take to choose faculty. And reading through the names, I saw Dr. Reed Carroll, whose name I recognized from the papers on synaptic plasticity. He has been studying AMPA receptors, and I thought it's good that Dr. Carroll is a relatively new faculty, so his lab will probably be not so big. He will not be so busy as more established investigators, and they're not always open to accepting new trainees, and their labs might be more competitive. So, I wrote an email to Dr. Carroll expressing an interest to train in his lab, and waited for a few days. There was no response. I decided to call. Emails get lost sometimes. Dr. Carroll said, "I'm sorry, I can't accept you in the lab." So I thanked him, and when the conversation ended, While I was going to open my computer and read my email, a voice came into my head. Well, on second thought, you have good experience. I can accept you to train. And I looked in my emails. Indeed, there was an email from Reed stating this, that he thinks my experience in biochemistry and molecular and cellular biology is good, so he can accept me. I was very happy, and we discussed the project based on his R01 grant, which was to study the different mechanisms of AMPA receptor trafficking internalization during long-term depression. Two forms, one induced by activation of NMDA receptors and the other induced by activation of metabotropic glutamate receptors. The project designed was to study a protein that interacts with AMPA receptors, GRIP. And I applied, waited a few months, and was so happy to find out that I had the fellowship. And when starting work as a fellow in Dr. Reed Carroll's lab at Albert Einstein College of Medicine Department of Neuroscience, I decided that besides learning the techniques for culture of primary hippocampal neurons in AMPA receptor internalization, I have to learn as much as possible about synaptic plasticity and read about this new area and attend relevant meetings. And in this process, I found out that protein synthesis is very important for metabotropic glutamate receptor long-term depression, and that it is enhanced in Fragile X, and I thought it will be very interesting to study protein synthesis, and one protein that is excessively synthesized is MAP1B. Why I thought it's interesting? Because it interacts also with GRIP, which we are studying, which is involved in the trafficking of AMPA receptors, and also it is part of the cytoskeleton, and the cytoskeleton is involved in trafficking of AMPA receptors. I discussed this with Reed, and he agreed. While designing the initial experiments, I indicated that I would like to have hippocampal neurons treated with DHPG, treated with NMDA, and control neurons, and to measure the levels of MAP1B. But Reed said, we don't have to have neurons treated with NMDA. We know that NMDA receptors don't induce protein synthesis. But I told him that it's good to ascertain in our system and for scientific rigor to have this control. So Reed agreed. I did 1-2 experiments, and I noticed that after activation both with DHPG of metabotropic glutamate receptors or with NMDA, there is increase in the protein levels of MAP1B is determined immunocytochemically. And in fact, I did a lot of these experiments which are here in my picture. Immunostaining of hippocampal neurons, and then observing under the fluorescent microscope. And I discussed these results with Reed and told him that I want to study protein synthesis. And in the meantime, I had looked up, are there any instances when activation of NMDA receptors in brain induces protein synthesis. And I found a paper by Alan Sheets, Angus Nair, and Martha Constantine-Patton that activation of NMDA receptors in the superior colliculus induces protein synthesis synaptically early in development. So I thought this is very interesting because MAP1B is expressed early in development at higher levels in Fragile X as a neurodevelopmental disorder. And moreover, this mechanism, which was described in the superior colliculus, was dependent on eukaryotic elongation factor 2 and its kinase. And I was acquainted with this kinase from earlier from my research in Dr. Ben Weiss's lab because I studied calmodulin, and this is a calmodulin regulated enzyme. I was going to work for Dr. Bill Hyde, and Dr. Hyde had described to me this kinase which has applications in oncology, and I was going to study it. So I wanted to study protein synthesis EF2 kinase, and I discussed with Reed, but he said that he wants to stay away from protein synthesis. I explained my rationale, but he still didn't think it's a great idea. So in the end, Reed said, if you find just one person in the Department of Neuroscience who agrees that your idea is worthwhile to pursue, I will agree. And we walked across the hall to talk with Dr. Scott Nowie, and Scott decided that my idea is good. And so I studied the role of MAP1B, which is induced after activation of NMDA receptors or metabotropic glutamate receptors in hippocampal neurons in the internalization of AMPA receptors, and we found out that this is through an EF2 kinase mechanism in an interaction with GRIP. I presented these results at the Cold Spring Harbor Laboratory meeting in 2006 on synapses, and later at the Gordon Research Conference, and they were published in the Journal of Neuroscience, which became my most cited paper. And interestingly, Dr. Kim Huber, who is an expert on Fragile X and synaptic plasticity, told me that she had also repeated my experiments. There was a paper published by a group at Johns Hopkins of Dr. Huganier, which also found the role of EF2 kinase in Fragile X and in mGluR LTD. And our paper in Journal of Neuroscience has been cited mainly because of the MAP1B EF2 kinase and protein synthesis mechanisms. So, as a conclusion, I would like to say that it's very important, I think, if you believe in something when doing science, to follow through. If your intuition is telling you to study something, it is probably important, so try to stand your ground. Another conclusion is that you have to read in detail what has been done and to know what the experts in the field have done so that you know what are the outstanding questions and what to study. And I have to say that you have to believe in yourself, because when I started looking for this fellowship, My mentor had told me that if I don't do residency, I'm out. I don't know exactly what he meant, but I think he meant that I can't be a scientist anymore. But I believed in myself and read about synaptic plasticity and applied for a fellowship and had worthwhile results, which now might be important for development of treatments for neurodevelopmental disorders. And it is also of note that this form of long-term depression is important not only for neurodevelopmental disorders, but also for addiction and for other disorders of learning and memory, such as Alzheimer's disease. I hope this story is interesting and helpful. Thank you.